So the IKI assay will tell us about the activity on the reactant side of our reaction. And the Benedict assay will tell us about what's happening on the product side. This will become more clear as we perform the experiment. Then we're going to be understanding the locking key formation, that there's a very important specificity property that enzymes have. So lipase will digest lipids, and amylase will digest carbohydrates, and maltase will digest maltose, breaking down maltose into glucose, and lactase is going to break down lactose into galactose and glucose. And then you also have sucrase. It's an enzyme that would break down sucrose into glucose and fructose. And then you have your proteases and peptidases, and these are enzymes that are highly specific for protein. So what we're trying to appreciate is that we have enzymes for every particular type of macromolecule or macronutrient. If you are a lipid, we have lipases. If you are a carbohydrate, we do not digest you with lipases because lipases have specificity for lipids, and peptidases have specificity for proteins, and amylase has specificity for carbohydrates. So if you put amylase with lipids, you will see nothing in the reaction because it's not specific. So that's what we're trying to appreciate, that amylase is specific for carbohydrates. And then we're also going to be learning that how bacteria might aid in digestion. So bacteria are equipped with built-in enzymes that are able to digest cellulose, for example. We are unable to digest cellulose, but the bacteria that happens to be living in our GI tract, having a symbiotic relationship with us, that bacteria is able to digest that cellulose. So let's learn more. Okay, so in this activity, you will investigate the specificity that enzymes have for their substrates. And we're going to be hydrolyzing or breaking down. So we're going to be hydrolyzing is when we use water to lyse, to break down. So we're going to add H2O, OH on one side and H on the other side to separate a bond between two connected units. So here we're going to be hydrolyzing starch, and we hydrolyze starch into maltose and maltotriose. And then the next step, maltose, as we said earlier, with an enzyme known as maltase, is going to break maltose for us into glucose and glucose. But the enzyme that will do the initial step that breaks down starch into maltose and maltotriose is known as salivary amylase or lingual amylase. That's in our mouth. So maltase cannot break down starch. Maltase will take it from here after amylase breaks down starch into after amylase breaks down starch into after amylase breaks down starch into maltose and maltotriose, then maltase will come in play. Okay, so the substrate is what we are taking into the enzyme to break down. So the substrate for amylase is starch, and the substrate for maltase is maltose. So the substrate is a substance that the enzyme acts on, and it has a pocket active site in which the substrate must bind, and these are non-covalent weak bonds, such as the ionic bond and hydrogen bonds. And you can read through that, but some questions on the pre and post lab will talk about what kind of bonds are on the active site of the enzyme. Are they covalent or non-covalent? So these are weak bonds, non-covalent bonds. Okay, so because the chemical changes that occur as the starch is digested to maltose cannot be seen by the naked eye, you need to conduct an enzyme assay. So we will conduct an enzyme assay. The chemical method of detecting the presence of digested substances You would perform 2 enzyme assays on each sample. So let's learn more about these assays. The IKI assay, and it detects the presence of the reactants in this experiment, which is the starch. So we are reacting starch with our enzyme amylase to break it down into our product maltose and maltotriose. And then Benedict will tell us how much maltose and maltotriose is there. So the IKI will tell us how much reactants do we have, the starch, what we are trying to break down. But the Benedict will tell us what's happening on the other side, on the product side. And that tells us these reducing sugars, known as the glucoses or maltoses, which are the digestion products of starch. So now these are the colors that will tell us. So not only do we need to understand what the assays are, we need to understand the color changes. So IKI is normally caramel color, but it will turn blue, black in the presence of starch. So after the reaction finishes, if you have a dropper that has IKI in it, IKI solution, and you drop it into the solution, if it is caramel colored, that means there is no starch. But if there is starch lagging behind that has not been in the solution, it'll turn blue. If there is a lot of starch, it'll turn black. So if one solution has blue turns from caramel color into blue, it has a little bit of starch. Or if it turns from caramel color into black, it has a lot of starch. So that shows you maybe amylase wasn't even there in that reaction, or maybe amylase is not working. It has been maybe temperatures too hot, denatured amylase, so amylase couldn't do the job, or the pH is not an optimal pH for it to work. So that is IKI, telling us what's happening on the reactant side. And there's no reaction that is only on the reactant side. We need to know what's happening on the product side. So on the product side, you have the Benedict reagent, which is naturally, just like IKI happen to be caramel colored, that turns on to blue and black. Benedict is not caramel colored. Benedict is actually blue, bright blue, but it changes it into green orange. So it turns into green, orange, or red. So the more maltose you have, the more you're going to be red. If there is mediocre amount of maltose, you're going to be orange. If there's a little bit of maltose, green. If there's no maltose at all, it's going to remain that color, bright blue. So it is important to understand the enzyme assays. Okay, so starch is a polysaccharide that is found in plants where it is used. It's a storage. It's a functional polysaccharide. It stores energy. Plants also happen to have another polysaccharide known as cellulose. It's a structure, it's not a functional. So starch is a functional protein. Its function is to store energy to be used later on. But cellulose is not used to store energy. It is used for tensile strength as a skeleton, backbone for the plant cell wall. So that is cellulose. But both of them are polysaccharides, and they are polymers of glucose, but they are structured differently. But the glucose molecules are linked differently, as we said. You will be testing salivary amylase, also known as lingual amylase, because salivary means it is part of saliva. But where is saliva coming from? It's coming from the lingual gland. So that is salivary amylase that is secreted by the lingual gland, known as lingual amylase or salivary amylase. They determine whether or not it digests cellulose. So we're also going to be testing on an experiment whether or not this amylase, we know amylase is strong enough to break down starch into maltose and maltotriose. But now we're going to be testing whether or not salivary amylase is able to break down cellulose. And obviously it will not, but we will test later on if bacteria is able to break it down. You will also investigate whether the bacterial suspension can digest cellulose, as we said, and whether the peptidase, and ending with ACE, it cuts. So peptidase cuts peptides. Maltase cuts maltose. Lactase cuts lactose. Sucrase breaks down sucrose. A pancreatic enzyme digests peptides, can break down starch. So it can, so this peptidase can break down, it's pancreatic enzyme that break down peptides. But that means it's specific for peptides. That means we hypothesize it will never have anything to do with carbohydrates. Okay, so these are the equipment that we're going to be using. You can read them at your own pace. And now let's go on the pre-lab. Which of the following is true of the enzymes and substrate? So if the product catalyst is present, the enzyme is not acting on a substrate. That's not true. Enzymes are specific about the substrate they act on. That is true. The enzyme is acted upon by the substrate. The substrate formed a covalent bond. No, it does not. But that is not true. If the product catalyst is present, the enzyme is not acting on the substrate. That is not true. The enzyme is acted upon by the substrate. Enzymes are specific about the substrate they act upon. That's true. Which of the following is or reducing sugar? So these are breakdown products. So maltose is broken down from starch. That makes a lot of sense. Okay. Glucose and maltose. That sounds good. Okay. Cellulose and starch are both found in the cell walls of plants. It's not true. Are polymers of glucose and found in the cell walls of plant. Polymers of glucose and polysaccharides. Proteins and peptides are formed by joining amino acids with a special type of covalent bond known as a peptide bond, which is the phospholipidases break down the proteins. We talked about that. The Benedict assay tests for the presence of the products, and these are the reducing sugars. Okay, submit that. Okay, now what we're going to do is we're going to drag a test tube on the incubator, and we're going to put different solutions to test. So let us go with the first two. So now not only are we dropping the content, but we also have to make hypotheses as we're going along. So the pH 7, that pH 7 buffer approximates the pH in the mouth. So in our mouth, the pH is 7, in the stomach, the pH is 2, and in the intestine, in the pancreas, the pH is around 9. So now let's begin. Tube 1 has amylase. Where's amylase? So 1 drop of amylase. And it also has starch. And it has pH buffer 7. Now that sounds like a perfect environment for me. I have my enzyme amylase, I have my reactant starch, and I have the optimal pH. So now I should see the perfect reaction happening in 1. That's my hypothesis. I should see a full reaction happening in 1. Tube 2. Amylase. Let's put 1 amylase and let's put glucose. So glucose is the product. So now I put the enzyme and the product and the optimal pH, but no reactant. So that does not make any sense for me. So I do not expect any reaction to be happening. And I should only expect to see glucose. And that means the Benedict reaction is going to detect for me that there is a lot of glucose. But in 1, we're going to be seeing very little. In the IKI, we should see a little bit of that natural color because everything has been converted into products. And we should see very heavy, like reddish color that because we have really became converted into products. 3, amylase, cellulose, pH 7. Amylase, cellulose, pH 7. And then tube 4. Tube 4 has for us cellulose. So in this one, amylase, in tube 3, amylase, cellulose. I have the proper pH, but I have something that I'm trying to see if amylase can digest. So I no longer have starch, I have cellulose. Let's see if that's going to work. And I hypothesize it will not because it's only specific for starch. Number 4, cellulose and pH 7 and deionized water. Well, isn't that interesting? I don't even have an enzyme here. I have a reactant and the proper pH and water and no enzyme. So I should see no reaction whatsoever for 4. Number 5, I should have peptidase. That's a enzyme that breaks down peptides. And I have my reactant as a sugar. So I should see no reaction because peptidase breaks down peptides. And starch is a polymer, not a peptide. So no reaction, even if I put the proper pH and the proper temperature. Lastly, let's put in bacteria with all of its amazing unicellular enzymes that it's equipped with. And then I have my cellulose, and I have the proper pH, but I don't have an enzyme, but I don't have to worry about that because I know the bacteria has all the enzyme inside it. Now, let's incubate. Incubate, we gently mix everything evenly. So you included tube 2, that is amylase, glucose, pH 7. To do what? To make sure the enzyme amylase is working. To see what a positive Benedict test. So that sounds for me like a positive Benedict test, because I cannot say there is no contaminating amylase, because I do have amylase in it. So that's not true. And I can't say I don't have contaminating glucose, because I do have glucose. This sounds like a positive Benedict reaction. So that Benedict tests for me whether or not I have reducing sugars like glucose. So when I add the Benedict reaction, it should give me a positive result that there is glucose. So that sounds like a positive control. So now let's incubate. So now we have incubated everything, and the timer is going down. We're evenly and gently mixing everything, and the timer has went down. And and then what we need to do, press next. Do you think test tube 3, amylase, cellulose pH will show a positive Benedict test? I do not, because I don't think that amylase can break down cellulose. Amylase can break down starch only. So I should say no. Do you think tube will show a positive Benedict? No. Okay, so now let's see that. Let's drag each tube. So drag the first tube in the incubation unit on the left side. And then, so we're going to take a little bit of each tube that we are testing, and we're going to see. So after the assay cabinet opens, notice the 2 reagents in the assay, the IKI. So we have the IKI to test our reactants, how much is how much reactant we have, and how much product we have, which are the digestion products of starch. Below the reagents are 6 small assay tubes into which you will dispense a portion of the incubated samples, plus a drop of IKI. Okay, so we took a little bit, we put a little bit of our solutions in this incubator, and then we're gonna run both the Benedict on this side and IKI on the other side. Okay, drag the IKI to the first. So now let's put IKI. Now let's notice how the color will change. Next. Now inspect the tubes for color change. So we said that a blue-black color indicates a positive starch test. So that means if we have a blue-black color, as in here, 5, that means we have starch. That means we had no reaction. So we have no products. That means we only have reactants. So our reaction did not go, our airplane did not take off. Everything is just in there. No reaction happened. So if starch is not present, the mixture will look like diluted IKI, a negative. So that's a negative starch test. The intermediate starch amounts result in a pale gray color. So if we have pale gray color, that shows there's an intermediate amount. And the IKI cellulose results in a negative starch. So I believe that this has a negative starch. So let's record the data. So we have recorded our data. We will inspect it later on. Okay, so which tube is included to detect contaminating amylase in the buffer or in cellulose? So if I want to detect whether or not there's amylase, I must ensure that the tube has no amylase. So how can I test if there is contaminating amylase and I already have amylase in the tube? So 1, 2, 3 doesn't make sense. It has to be, so cellulose, so which tube is included to detect contaminating amylase in the buffer or in cellulose? So this one tells me that there is no amylase, so that sounds for me, so none of the above. Which tube is included to detect contaminating amylase? So this has no amylase, and in the buffer or in cellulose. Okay, so that means we needed to have a tube that says pH buffer, deionized water, and then test it for amylase contamination, and then a separate tube that says cellulose and deionized water, and test it for and test to see whether or not we will have products indicating that there is so if the reaction goes on, we will see that there is contamination. So here, if I have cellulose and pH buffer and deionized water, that will tell me whether or not, if I see that there are products, that means there is a little bit of amylase. So amylase does not digest cellulose. So that's not true. I need to have starch here. So that's the mistake. We know that amylase will never digest cellulose. So we can never test if there is amylase in the buffer or in cellulose by using cellulose. So we need to put starch. So if we have starch, pH 7 and deionized water, and then we see that there is a color change in the Benedict trial, that will show us that the starch has been converted into its reducing sugars. So that means there is some hidden amylase contaminating. So, but we cannot use that for cellulose. So the right answer is none of the above. I'll talk about that more in class. Okay, drag the Benedict reaction dropper cap here. So now we're looking for products. We're looking for products. Let us look for products. Select boil, the entire rack descended, and then we have our results. If you remain blue, that means you had no product. But if you change, that means you did have a reaction and you have reducing sugars as products. Next, let's do record data. Now, what is the usual substrate for pancreatic enzyme peptidase? We know peptidases are specific for peptides. Come on now. Okay. Now let's submit. We have submit there. Okay. Now let's look at the post lab. Now let's first look at the experiment. So in tube 1, we had amylase. That is our enzyme, and starch. And that is the optimal pH. So in the optimal pH, so that means you'll have a full reaction where reactant starch is going to be converted into product reducing sugars, maltose and maltotriose, glucose. And the temperature is optimal, the pH is optimal. So that is a perfect result. I have zero, I have no IKI. That means all of or most of my reactants have been converted into products. So that's perfect, number 1. For 2, I do have my amylase as an enzyme, but I do not have a reactant. I have a product, but amylase works with reactants, converting them into products. It does not work with products, converting them back into reactants, even though I do have my proper pH and my proper temperature. So that shows me that when I do my Benedict will tell me there is glucose. So Benedict tells me there's glucose, but I have no reactants because I started with products. So I tested here. I began with reactants that have been converted into products. That's why Benedict is positive. But here I began with products and I detected them as products. So I had no reaction here. 3, amylase and cellulose. I did not detect any reactants because IKI detects starch. and Benedict detects reducing sugars. And cellulose is a polymer. So I cannot detect anything because I do not have my reactants for AKI, and that's the starch, and I do not have products for my Benedict, and that is reducing sugars. So I should have null for both. Cellulose, you see, whenever I have cellulose, no reaction will unfold. Here, I didn't even have an enzyme. Peptidase. So for peptidase, given peptidase starch, I was able to detect with the IKI that I do have, so apparently IKI can detect if there are there are proteins or glycoproteins, proteins that are mixed with sugars. So that's why it went positive. But obviously, I do not have products because peptidase cannot convert reactant starch into reducing sugars. Now, this is interesting. So bacteria, I had the bacteria and cellulose with the proper pH, and now I do have Benedict reaction, and that shows us that we succeeded converting cellulose into reducing sugar. So that shows you that bacteria does have the proper enzymes to break down the bonds between the cellulose polymers and digest it into reducing sugars. Okay, substrates for amylase is our starch, obviously. Results from test tube 5, peptidase starch, demonstrated that peptidases cannot digest starch. Why was cellulose in tube 6 that has bacteria, cellulose, hydrolyzed glucose? The bacteria suspension contained enzyme amylase, which digested cellulose, which contained an enzyme peptidase, which is no, it's contained an enzyme cellulase. There you go. Cellulase breaks down cellulose. Which tube allowed you to conclude that the buffer was not contaminated with amylase? So if I want to ensure that there is no amylase contamination, that means the tube is not supposed to have amylase. So those three are wrong. So the answer is one of those three. So which one shows that there is no contamination with amylase? I know that cellulose can never be broken down by amylase. So here I can have some amylase contamination and I still will have no product. So that's not true. So the answer is either 5 or 6. Peptidase starch. Peptidase was not contaminated with amylase. I know amylase cannot break down cellulose. Only cellulase breaks down cellulose. So here, so peptidase and starch. So when I have peptidase and starch, I'm supposed to get nothing, 0, because peptidase is specific for peptides. But if there's contaminating amylase lagging behind in the test tube that I assumed only has peptidase and starch, what will happen is I will have some reducing sugars formed as products. So this is definitely the right answer. Submit. Okay, describe why the test tube 1, amylase, starch, pH 7, and test tube 2, amylase, glucose, pH buffer are the same. So why is test tube 1 and 2 the same? We talked about that. For 1, Benedict will only be activated when there are reducing sugars. For 1, I began with a polymer that was broken down by amylase into reducing sugars that were detected by Benedict. That's why we have a positive Benedict. But for 2, I have amylase. Amylase that did not do anything because there are no reactant polymers. We only have products. So Benedict will never know whether the glucose it's detecting was a product of amylase breaking down starch, or it was just, so if you add Benedict to a test tube that only has glucose, it will turn plus, because all that it does is that it turns glucose. It detects glucose, but it will never detect why was the glucose there. Was it there because of an amylase reaction? Or so if you have a test tube that you just poured glucose in it, test tube number 1. And you have test tube number 2 that you had starch plus amylase, and you drop Benedict in both, Benedict will detect glucose because that's all that it does. It just detects glucose. So that's why they look the same. Describe the result in test Tube 3, amylase, cellulose, and pH 7 buffer. How well did the results compare with your prediction? So amylase and cellulose, I'm supposed to get nothing. Amylase, that's tube 3, amylase, cellulose, I'm supposed to get nothing because cellulose is only broken down by cellulase. So do you think this tube 3 would show a positive benefit? No, and that is because cellulose cannot be broken down by amylase. Amylase only breaks down starch. Cellulose is only broken down by cellulase that is found in bacteria. Describe the usual substrate for peptidase. These are peptides. Lastly, explain how bacteria can aid in digestion because they have these very unique enzymes known as cellulase, and they help us break down the cellulose. Press submit. These are that's the only wrong answer that we've gotten because the contamination with amylase can never be detected with cellulose because amylase does not even digest cellulose. So we needed to put starch, for example, to see if amylase is there or not. Okay. And that sounds like the end of our session. See you in our next experiment.